So I backed up a couple slides from where the lecture left off. Just to show this particular diagram, if you had lab, this should also look very familiar. It is one of the ways we really try to help sort out different bacteria into different genera, into a different genus. If you have a gram-positive cocci, there are lots of bacteria that are gram-positive and cocci-shaped. Most of them even have the word coccus in their names. And so we can do several different lab tests to help sort and narrow them down. Everything before this is all about the staph. And then if you have a catalase negative test result in lab, we know it's either going to be a Streptococcus or an Enterococcus. And I started in the recording talking a little bit about the Streptococcus, specifically the Streptococcus pyogenes, the bacteria that causes strep throat. But it's not the only Streptococcus bacteria out there. So I'm going to kind of pick up where that lecture left off, talking about some other Streptococcus bacteria. There's enough Streptococcus bacteria that we have different groups of bacteria. Streptococcus pyogenes, the one in our lab that we were trying to identify to see if you were carriers of, were in the group A. That's why we even had that selective group A plate. But I want to get to where the lecture left off, which I think was right here, talking about things that we even did in lab, how to identify strep pyogenes. But as I just mentioned, Streptococcus pyogenes is not the only strep out there. And again, most of the bacteria we talk about in here are all going to be the most common bacteria that cause disease. They're not the only bacteria out there. Like, there are so many more bacteria that our class cannot cover because it's not strictly a bacteriology class. So we just go over the most common ones and the most common that are going to cause disease that you may encounter in your future careers. A bacteria that's in the group B Streptococcus group, again, there's lots of strep, but there's lots of groups, is called Streptococcus agalactiae. It, as its name implies, it is a coccus-shaped bacteria. It's gram-positive, and it does like to hang out in chains. So not all Streptococcus hang out in chains, but this is one that really does like to. It's different than group A is because on the outside of it, it has different antigens. It has B antigen, puts that in the group B of Streptococcus. Most of the group A do beta hemolysis. That's what we were looking for on your plates to, give us a clue if you had strep pyogenes. This does a beta hemolysis, but it's a lot harder to see. Really the stab marks are almost going to be absolutely necessary to see beta hemolysis without turning all the lights. Maybe I can. Let's see. It's still almost hard to see some of the beta hemolysis on the plate. Doesn't want me to turn all the lights off. Maybe. There we go. It is also resistant to the antibiotic bacitracin. So bacitracin is very effective for strep pyogenes, but Streptococcus agalactiae is resistant to it, which means if you're resistant to an antibiotic, you will, it will do absolutely nothing. The bacteria will grow right up to the antibiotic because that antibiotic has no effect on them whatsoever. And so knowing what antibiotics are useful or effective against different bacteria can actually help us identify the bacteria. And then we know how to treat it as well. Now, where Streptococcus agalactiae causes the most issues. The most issues is usually neonatal. So when pregnant women are carriers of this particular bacteria, they pass the bacteria onto their infant in childbirth. And so it can cause bacteremia, that's bacteria in the blood. It can cause meningitis. It can cause pneumonia in newborns. I mean, poor things just got born and now all of a sudden they're sick. This happens on average 3 out of every 1,000 births. And it can cause enough, bad enough bacteremia and meningitis. It can cause blindness in infants. It is a concern. Now, it also can cause issues in immunocompromised patients, but the most cases that we see of it are newborns, is that neonatal disease. The best prevention, I'm going to skip ahead, the best prevention to prevent that is mom takes antibiotics before childbirth. they do test mom to see if they're a carrier of this bacteria before childbirth, so that they know they can almost prep for it and actually put mom on antibiotics before childbirth to try to make sure to kill them. Even though they might have no issues whatsoever on mom, mom's a carrier and passes it on to the newborn suffers. To diagnose this particular bacteria, they can do an ELISA that's looking for the particular antigen using those enzyme-linked antibodies that gives a nice little blue color change. And it's super treatable. We have antibiotics that treat it, no issues. The biggest issue is testing for it to know that you can then treat for it. So again, it's not a common, this is not something that I would expect you guys to have, possibly carriers, but not suffer any serious side effects itself. Again, it's usually more tied to just those neonatal childbirth cases. This bacteria, you guys probably all have though. These are Streptococcus bacteria. They're what's known as the Viridans group. They're not group A, they're not group B, they're not group C. They don't have any of those particular antigens. There are so many strep bacteria out there. They're put into different groups, but then we even have a whole separate group called the Viridans. So it's like its own unique group. It doesn't get the A or B qualifications, it just gets its own separate group of kind of the oddball bacteria. Now, most of these bacteria are normal oral flora. So when we did your throat cultures and you held your plates up, you all saw some greening on the plates. It was going to most likely be some type of Alpha-Hemolytic that gave that green color, Streptococcus bacteria. A lot of normal oral flora, so it likes to live in your mouth. They only become issues is when they get somewhere where they're not supposed to be, and usually for those that are immunocompromised. So these are bacteria that are in your mouth that if you don't brush well enough, they're the ones that are eating all the sugar that you feed them. They're producing acid, and they can lead to dental cavities. They can build up and form dental plaque on your teeth. And if you floss and you have weakened immune system, if you floss or you scrub too hard and all of a sudden you have bleeding gums, those bacteria can get into the bloodstream. Now, those that have a healthy immune system can clear it. Those that don't have healthy immune systems, and now you just put bacteria into your blood, it can cause meningitis or endocarditis. That's inflammation of the heart. So those that are immunocompromised, before they go to the dentist and have any kind of dental procedures, are usually given some type of antibiotic. It's just a prophylactic antibiotic. Those that have diabetes, that puts them at immunocompromised state, usually given antibiotics before they go to the dentist, because the dentist is poking and prodding and lots of potential openings for putting these bacteria into the bloodstream. One particular bacteria that is in the Viridan group that can cause more issues is Streptococcus pneumoniae. Still, it's gram-positive, still cocci-shaped, so we're still in that whole group. They usually like to hang out in short chains, sometimes in pairs. Also, Alpha-Hemolytic. It also is not a group A or group B. It doesn't have those Landsfield antigens that puts them in the A's or B's or C groups. It's also in this kind of random group of Viridans. So it's kind of in the still oddball group of Streptococcus bacteria. But it has a lot of virulence factors, meaning lots of things that help it cause disease. And the more it has, the worse off for us. One, it has a capsule. And we've already talked a little bit about capsules in lecture and in lab. Having a capsule gives that bacteria a little bit of camouflage, because the bacteria can't recognize it right away. It can help it stick to surfaces to call home. Our phagocytes can't quite get around it and break it down. All good things for the bacteria. It also makes a protein adhesion to help it stick, because before bacteria can reproduce, they first have to find a home. They can make an enzyme called secretory IgA protease. It's an enzyme, it ends in ACE. There were 5 different antibodies. Remember, we just got done with our immune system. There was IgA. There was IgM, IgG, IgD, what am I forgetting? IgE. So we have immune system cells. We have antibodies that try to bind bacteria and target them to get rid of them out of their body. That's the purpose of our antibodies. This particular bacteria makes an enzyme that destroys our antibodies. So our immune system response is to make antibodies and target these things to get rid of them. This bacteria is like, ha ha ha, I'm just going to go ahead and kill those antibodies and destroy them so the antibodies can't do their job. It's great for the bacteria, bad for us. They also make a chemical called pneumolysin that breaks apart epithelial cells. It allows this bacteria to travel and get into new areas in the body. Again, it's all good things for the bacteria, all bad things for us. Now, it is called Streptococcus pneumoniae, and yes, one of the things that can cause, based on its name, is pneumonia. It's not the most common cause of pneumonia, but it is one of the causes of a bacterial pneumonia. People that are at highest risk for getting the pneumococcal pneumonia, elderly, immunocompromised, and infants. So you will probably hear the word pneumonia a lot, and there are lots of things that cause it. Pneumonia itself, how do I say it, is not a disease, because a disease is caused by a specific organism. Pneumonia is a condition. That just means you have fluid in your lungs aren't doing what they're supposed to be doing, but there's lots of things that can cause it. And so we have to narrow it down. So we call it a pneumococcal pneumonia. That coccal part is because it's from a cocci-shaped bacteria. So that's the big clue that it's a bacteria that's causing this pneumonia. And it's a cocci-shaped bacteria that's causing the pneumonia. It's not a virus, and it's not any other type of bacteria. But Streptococcus pneumonia can get other places in the body and cause more common diseases. It is a big cause of sinus infections. That's the sinusitis. It's a big cause of inner ear infections or middle ear infections. That's your otitis media. So ear infections and sinus infections, top causes, they're caused by Streptococcus pneumonia. It can, however, get into the bloodstream, causing bacteremia. It can get in by the heart and cause endocarditis, and it can cause meningitis if it gets up near the brain as well. So it's a bacteria that, although its name says pneumonia, it is not the only thing that it can cause. Really, you'd say there's more cases of ear infections and sinus infection caused by this bacteria than anything else. Now, to diagnose it, if they suspect that it is causing a pneumonia, they're going to gram stain a sputum sample. So they're going to collect a sputum sample. The patient coughs up a nice big old good sputum. They can gram stain it and look directly for the bacteria. They can also culture it out. They can look, what kind of hemolysis does it have. It also has that nice alpha hemolysis. They can look to see if it's sensitive to the antibiotic optogen. We had this up in lab. We had a Streptococcus pneumoniae with a little optogen disc on it. Treatment, still treatable. Penicillin's the top treatment. If it's becoming resistant to it, as some strains are, then we switch over to erythromycin, still quite treatable. Benefit though, or a bonus, is we do actually have a vaccine for Streptococcus pneumonia. I think I just last week saw a commercial. I think the first time I've seen an actual commercial advertising this particular vaccine, because the ones that are going to suffer the most severe side effects, elderly and immunocompromised, and so it was a vaccine. Now you're going to watch for the commercial, you'll notice it. When you do, it was, elderly people, I'm getting protected, blah, 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 against the streptococcal pneumonia vaccine. So there is a vaccine out now. I've never seen it advertised till like a week ago. All right, back to kind of where we're at. So we've got gram-positive cocci. If they were catalase positive, it's a staph. If catalase negative, it's either a Streptococcus or an Enterococcus. So we've already talked about some of the Streptococcus bacteria. Now we're going to move on to the Enterococcus. Now, anything that has entero in its name implies intestines, or GI. So these are bacteria that are going to be more normal flora of our intestinal tract. But they are still gram-positive cocci, and they're still catalase negative, just like strep. So when we gram stain them, they're gram-positive. They usually like to hang out in short chains or pairs. Again, gram staining gives us a lot of information to help narrow bacteria down, but there's still a lot of bacteria that they share similar gram stain results. There are two main species of Enterococcus that cause human disease. Lots of other Enterococcus out there that don't cause us disease, so we don't care about them. But the two that cause human disease, Enterococcus faecalis and Enterococcus faecium, they normal flora of the human colon. They only become issues is if they go somewhere where they're not supposed to be. So anytime they go somewhere else, it can cause urinary tract infections. It can cause skin infections. Again, that just means you're going to have fecal matter that gets into open wounds. But they are a top cause of healthcare-associated infections. Nosocomial infection is a healthcare-associated infection. Most healthcare-associated infections are fecal-driven, just FYI. There's just fecal matter everywhere, and if there's any type of medical procedure that has to do anything with the intestines, that just opens that possibility of fecal matter and bacteria getting into lots of areas. Now, to diagnose it, these bacteria are not sensitive to bile. Now, if I go back, Streptococcus bacteria are sensitive to bile. Now, sensitive to bile means they will not grow in the presence of bile. Enterococcus could care less about bile because they live in the intestinal tract. That's where bile is. So they'll grow in the presence of bile just fine. Streptococcus will not. It is one way we can narrow down. These are two bacteria that are going to gram stain the same, and they're both going to be catalase negative, so how do we start to sort them out? One is to see if they'll grow in the presence of bile. We do have media. We do have plates that actually have bile added into that agar, and we'll look to see if it grows or not. If it does, we know it's an Enterococcus. If it doesn't, we know it's a Streptococcus. So anything Enterococcus will grow in the presence of bile. It's used to it. It's evolved to it. Treatment is getting harder, though, because Enterococcus bacteria are becoming very resistant to our antibiotics. We do still have some that are working. It is not completely resistant. Prevention is difficult because it's part of the normal flora. So this is part of people's normal flora. So we know it's going to be there. We can't just get rid of all of it. Our only way to really prevent it is minimize transmission. Good hygiene is making sure hands are washed and making sure everything is washed, wearing proper PPE. It's anything just to minimize the transmission so the bacteria doesn't get where it's not supposed to go. Now we're going to switch shapes and we're going to look at gram-positive rods. Now, of all the gram-positive rod bacteria or gram-positive bacilli, we split those into two groups, whether they can make an endospore or not. And so we've already gone over and talked a little about just what an endospore is and the advantage of having an endospore. And there are two groups of bacteria, two genera that can make endospore, the Bacillus and the Clostridium. So we're going to talk about those guys first. And we're going to start with the Bacillus. So when we gram stain it, it doesn't always gram stain accurately if it isn't in an endospore state, but it will pick up the purple. Your best bet to know if it's in an endospore state, though, is to do an endospore stain. Really, the only bacillus that causes human disease is Bacillus anthracis. There are lots of bacillus out there. I even know the very first lab when you guys were swabbing random things, I could tell just based on what it looked like on a plate, there were some bacillus bacteria, not disease-causing bacillus. Really, the only bacillus we have to worry about is Bacillus anthracis, and it's really not even that common to even really start to worry about it. Now, it's not the bacteria that causes the issue, it's the toxin that the bacteria makes that becomes deadly. How we as humans generally pick up this particular bacteria are from infected animals. Because we hear anthrax talked about when do you normally hear of anthrax? Like movies, bioterrorism. Like that's when you're like, oh, anthrax. It is used as a bioterrorist weapon, very rare. It can be deadly. It's just we don't encounter them on a normal day-to-day encounter. How we can pick it up. There's 3 ways we can pick up this particular bacteria, and depending on how we pick it up, depends on what kind of symptoms you have. We can either ingest the spores, so you're going to eat the bacteria. They can get in through the broken skin, or you can inhale the spores, which that's usually the bioterrorism method, and it is the more deadly disease-causing way of picking up the bacteria. So the 3 ways that you can pick it up depends on which type of anthrax disease you now have. If you ingest the spores, you have gastrointestinal anthrax. It's extremely rare. I don't remember if I wrote down the numbers. I think it's like 1 case a year, maybe, even happens. It's extremely rare. I don't have a number. I was like, I feel like I looked it up at one point. It's deadly. About 50% of those that do get it die, but again, it's extremely rare that you would ingest enough of the bacteria that the toxin would kill you. The most common form of people getting this particular bacteria is if it gets into an open wound. If you were working with animals, again, they are kind of the main mode of transmission. If you're working with possibly infected animals and you have an open cut, that bacteria gets into that open cut and it causes what are known as eschars. It almost starts to kill the tissue where the bacteria is growing and you have these blackened lesions. That's the most common form. It still is not that deadly, and generally I would hope you would go to the doctor if your skin started to look like that, and it's treatable. Inhalation anthrax is if you inhale the bacteria, which again is extremely rare. Again, I think there's only maybe, I don't even know if there is a case a year. Like, I don't think we even have that much. It would have to be a bioterrorism type of infection. But the bacteria can get into the lungs, and as they start to grow in the lungs, they produce the toxin, and it destroys your lung tissue, and that's how you die. Extremely rare. It's not something you would normally encounter on a day-to-day basis. So it's a rare bacteria, but it is a bacteria that's in the bacillus that can cause serious disease. How they would identify it, well, they could culture it, they could grow it out. On a blood agar plate, bacillus of any type of species are usually going to be pretty big colonies, and they're usually going to be very white. A lot of them are going to do a beta hemolysis as well. So that's why I'm like, I saw some of that on some of your guys' plates in that first week. Big, huge colonies with a lot of beta hemolysis around it. Again, not anthracis. But it's treatable. We do have antibiotics that treat it. You just have to get diagnosed. Biggest way is, if you work with animals, we do have vaccines that can treat animals. So animals, we do vaccinate against the bacteria. And we do actually have a vaccine for people that work or at high risk. They work with possible infected animals. Because it's so rare, we don't get vaccinated for it. It's so rare that we would ever encounter it, but we do have a vaccine for those that are at a higher risk. The other big group of endospore-making bacteria are the Clostridiums. You guys are most familiar with which one? It's the C of C. diff. And so it is one of the big Clostridium-causing diseases. But there are 4 big Clostridium diseases that it is a guarantee in the healthcare setting or just in your normal everyday life, you will have experience with all 4 of these Clostridium. So C. diff is like the one you hear all about, but you will have some experience with all 4 in your futures. So Clostridium, when we gram stain it, it's an endospore. It's not going to gram stain accurately, but it will pick up that purple. It looks a little different than Bacillus. Bacillus like to hang out in nice long chains. Clostridium, not so much, and a lot of times they even will have, if they go into an endospore state, make it longer. Ugh. They like to go into their endospore state on one end. You can almost see like an open circle. Those are bacteria that are going into an endospore state. So they look a little almost club-shaped. They are strictly anaerobic bacteria. So they will only grow in anaerobic conditions. And that's huge, because if you're trying to identify this bacteria and you're trying to grow them in a lab and do different testing procedures on them, everything has to be done in anaerobic environments. Now, Clostridium, because there are quite a few species of Clostridium. There's multiple places you can find it. We can find different strains of Clostridium in soil. We can find it in water. We can find it in the GI tract of animals, and we can find it in the GI tract of humans. There's lots of Clostridium species out there, and so we can find them lots of different places. Some of its virulence factors that allow it to cause disease and cause huge issues with us is that it has digestive enzymes, so it will digest parts of us. It has lipases, that means it's an enzyme that breaks apart lipids. And it has proteases, that's an enzyme that breaks apart proteins. So it can break apart us from the inside out if it wants to. Now our first, because there's 4 Clostridiums. Our very first one is Clostridium perfringens. This is a bacteria that likes to get underneath the skin. It gets into an open wound, and it gets into the skin, into where it's an anaerobic environment. And when it grows, it produces toxins that causes irreversible damage to that tissue. Again, it has digestive enzymes. It starts to digest your skin from underneath. Now, as it does that, it does cause what's known as gas gangrene. And that's what the picture is that is a bacteria that's underneath the skin that is breaking apart the lipids, it's breaking apart the proteins, it's breaking apart the tissue. It's breaking apart that skin itself. As it does that, as it's growing just oh so happy, because it's happy, it does produce a very foul smelling gas. Has anyone worked with anyone that's had this? I was like, sometimes I've had students that they worked in nursing homes where they've had this, or they've worked in different hospitals or something that patients have had this. Because I have not worked with a patient that's had it, but everyone that says that they have, it's like, it's a smell you'll never forget. Like, it's extremely gross and smelly and very noxious. Again, the bacteria is producing a very foul smelling gas, but you also have the death of tissue. So you've got dead tissue now on the skin. Now, getting under the skin is probably and causing gas gangrene is going to be its top cause of diseases. But if we somehow ingest the bacteria as well, because it gets into food, and it grows in the food in an anaerobic environment, it can cause food poisoning as well. So how we diagnose someone, to say that you have Clostridium perfringens and that you actually have an infection caused by that, we look to see how many bacteria there are per gram of food or how many bacteria there are per gram of fecal matter. Just having a few bacteria is not enough to cause gas gangrene. It's not enough to cause food poisoning symptoms. We're like, you have to have this much bacteria to really actually say that's what's causing the infection. And it is quite a bit. If we grow it out in the lab, it's going to be hard to see without dark. It does a double zone of hemolysis, that right around the colony, there's almost a very distinct beta hemolysis that you could see right through it. But along an edge, I don't know if you guys can tell, it's a little bit redder. It's still a beta hemolysis, but it's not a complete clearing. So it does almost this weird, unique type of hemolysis. Treatment, there is no treatment for the food poisoning. And I'm like, the diarrhea, the vomiting will get rid of the bacteria all on its own. So just, replace fluids and electrolytes. Gas gangrene, though, again, this is death of tissue. The only way to get rid of the bacteria is to remove the infected tissue. So it could mean amputation. It could mean the actual, it's called debridement, where they literally scrape off all the infected skin and tissue. Mm-hmm.
I was going to say, I worked at a nursing home that we had a resident who had tissue dying. I guess I have witnessed this before. She had tissue that was black on her tailbone because she had this bed sore there. And we had to crack a window. It was so bad.
So it might have been this particular one. It's one of those, there might have been other ones too. And I'm like, but
yeah, we had to measure how deep it went and then clean around it.
Did they go in and surgically remove all the tissue? Because I feel like at some point that's going to spread. It was interesting because I had a student just last semester. She came in and she's like, I was working this weekend and I had a 20-some-year-old patient that had to have an arm amputated. And they're like, yeah, it was like some Clostridium bacteria. I'm like, perfringens? Yeah! And I'm like, I mean, it happens. I don't know how it got bad enough that they had to amputate part of her arm. But it is a very deadly bacteria. But normally the cases I see are diabetics because they already have low immune systems and poor circulation. But it's just a bacteria, you know. The best way is making sure all the wounds are cleaned properly, that you don't get this particular bacteria into that open wound. And then to stop the food poisoning, make sure foods are refrigerated properly. So it's one. At some point in your careers, I have no doubt you will experience this bacteria. Not like you, but your patients. The second Clostridium, the one that you are most familiar with, at least have heard of it, is the Clostridium difficile, also known as C. diff. Now, they have, again, tried to change its name. And so don't be surprised if you're like, that doesn't sound like Clostridium. They have changed it to Clostridium. I'm going to spell it. Clostridioids. It's like a weird name. Because there are actually so many Clostridium. We're going to go over the top 4 that cause disease that I have no doubt you'll experience at some point in your careers. But there's so many Clostridium out there, ones that cause human disease, ones that cause animal disease, ones that cause other issues, that they were trying to sort them out and organize it and rename them to divide them all up. And they actually wanted to call this particular one like Clostridium, but then everyone got real mad because they'd have to change all the C's to G's, and then it would be G. diff, not C. diff, whatever. They finally said, fine, we'll just make you happy and get rid of the gastro so that it's still technically C. diff. But don't be surprised as you start to see things, you may not actually call it Clostridium anymore. Because it is officially named now, so the newer textbooks will have it as Clostridioids. The older textbooks will have it as Clostridium. It's not like the bacteria change. They're just renaming things. But it is, for a lot of people, part of their normal flora. You guys might have C. diff in your intestines right now. It's not causing you any issues whatsoever. That's because your other bacteria are keeping the C. diff in check. That's why I'm like, it's good to have normal flora bacteria. There's only so much food, there's only so much space in your normal flora. Keep Clostridium difficile from growing uncontrolled. So it's an opportunistic pathogen, which means it will cause disease if something else changes, like a location. But this isn't a location. It will cause disease when you kill your normal flora. So those normal flora that keep the bacteria in check, if you kill them, well, now all of a sudden your C. diff don't have anyone to keep in check, and they throw themselves a little party, and that's when they cause serious diseases. So they can cause minor abdominal infections to explosive diarrhea. That's textbook written. The bacteria, again, it's not the bacteria that causes issues. The fact they make different types of toxins that can damage our tissue. They can get into the colon, and as they start to damage the intestine lining from the inside, we start to get ulcers, and then our body tries to cover them up. And so we almost start to get, it's called pseudomembranous colitis. It's ulcers inside of the abdominal cavity. But we run into an issue. If you have ulcers inside your abdominal cavity, this is also where fecal matter is. So now you can have bacteria getting into open wounds inside your intestinal cavity. It's going to be very painful, and it can cause lots of issues. Now, it is a nosocomial pathogen. It's a healthcare-associated pathogen, not because it's always picked up in a healthcare setting. It's because you suffer from it after going to a healthcare setting. If you went to a healthcare setting because you had, a horrible ear infection, sinus infection, whatever, and they put you on a broad-spectrum antibiotic, well, that killed all your normal flora, and then a week or two later, now you're having all these symptoms of the Clostridium difficile. It's because the C. diff are now having a party because you killed all your normal flora. So it's not that it's picked up in a healthcare setting that makes it a top cause of a healthcare-associated infection. It's because of the procedures, aka the antibiotics, that happen at the healthcare setting that causes it to have a party. It can get picked up, though, in healthcare settings, though. So if you weren't a normal carrier of it, doesn't mean you can't pick it up while in a healthcare setting. Now, to diagnose it, they're going to look for the bacteria from the fecal matter. They can look for the actual toxins and identify the toxins. They can do a PCR, which is going to look for the DNA of the bacteria. There's lots of different ways we can identify the bacteria. Treatment. It's one of the few treatments for a bacteria that instead of giving antibiotics, it's to stop taking antibiotics so that we can try to get our normal flora back. If, however, this bacteria is growing so uncontrolled and we don't think there's any chance our normal flora are going to come back and really provide any benefit, they will give a really strong antibiotic called metronidazole. But otherwise, the first thing they're going to do is just take you off your broad spectrum antibiotics. And one of the newest methods of treatment for people that have C. diff infections that they just, no matter what they do for treatment, can't get rid of it, is a fecal transplant. Have you heard of it? I was going to say it's new just in the last maybe 5 years. Is they know that normal flora bacteria will keep this bacteria in check. But if you killed all your normal flora bacteria, it's going to take a while to get them back. So what they can do is actually take fecal matter from someone else and put it into your intestinal tract, and their normal flora will grow in your intestinal tract and it will target and keep these guys in check. So it's just taking other fecal matter from someone else. It's one of those, the biggest issue right now, because it is starting to get used, it is starting to become more common. I don't know if I would say it is common yet, because again, they're literally looking for people that it's just not going away. The biggest issue though is the insurance companies are like, I don't even know how to code this. You know, is it an organ transplant? Like, what is it? To try to figure out, the insurance companies don't know how to code it for medical reasons. But it is extremely effective. Like people that suffered from C. diff infections for years that they were in pain, fecal transplant, cured. Like it's amazing. Now, best prevention, good hygiene, probiotics that try to keep your normal flora healthy. That's about it. Our third Clostridium bacteria is Clostridium botulinum. Again, it's not the bacteria that causes the issues generally. It's the toxin that causes the issue. So most people suffer from botulism when you ingest toxins. A lot of times the bacteria can die, but if you ingest the toxins that are still present, you now have botulism. So intoxication is because you ingested the toxins. That's where the word intoxication comes from, is you ingested toxins. Now, a little A&P review. You ready? It's like the nervous system, neuromuscular junction. How the nervous system causes your muscles to contract. So, if you've got yourself an electrical signal, it goes to the neuromuscular junction. This is the end of an axon. And that action potential is going to cause these little vesicles that have acetylcholine in them, that's the little blue dots, to release. It's going to go across that synaptic cleft. It's going to go into the actual skeletal muscle itself, and the skeletal muscle, here's the zoomed in picture, the skeletal muscle can contract and do whatever it needs to do. So action potentials, acetylcholine goes across that cleft, and the skeletal muscle can contract. That was like a full lecture in A&P in like 30 seconds. Now, what the toxin does, what the botulism toxin does, is it coats the end of those axon terminals. And so when that information comes down here that says we need to contract this particular muscle, that acetylcholine can't cross the synaptic cleft. It can't tell that skeletal muscle to contract, and the skeletal muscle will not contract. So this toxin prevents skeletal muscles from contracting. That's not good. Now, you have an extremely important skeletal muscle, and you're like, whatever, so I just can't do this. You have an extremely important skeletal muscle that's necessary for survival. Mm-hmm.
So that's Botox, right?
Yes, that's what botulism toxin stands for. So that's the idea of Botox, is if you can't contract your facial muscles, you know, if I do this, I know I've got wrinkles up there, but if I can't contract that muscle, oh, not as many wrinkles. But this particular bacteria, where was I at? No, I forgot. It's the, well, whatever. It's the toxin that kills you. So there's 3 ways that this particular bacteria gets into the body. Well, I shouldn't say get in. There's 2 ways it gets in the body, but there's 3 different types of botulism. There's foodborne botulism, that's the most common, infant botulism, and wound botulism. So foodborne botulism is if you eat food that this bacteria was in, and it was growing, and all of a sudden you just ingested a whole bunch of the toxins. Oh yeah, the most important skeletal muscle that we need for survival. It's not these arms. Your diaphragm is a skeletal muscle. If your diaphragm can't go up and down and contract, you can't breathe. And what happens if you can't breathe? You die. And so if your diaphragm can't contract, you die. This is an extremely deadly bacteria because it's an extremely deadly toxin that it makes. So most common way that people pick up this bacteria is eating improperly canned foods. Is that this bacteria got into the food, and then if you canned the food and it was left on the shelf, it's an anaerobic environment, and they were sat there and just continued to grow and make toxins, and then you ate the food and it had the toxins in it. You now have foodborne botulism. How you would know you have foodborne botulism? Usually within the first day or two, it already starts to target certain muscles. Usually the smaller the muscles, the first. Usually it starts to target the eyes and the pupils, and all of a sudden everything becomes very blurry. You can't dilate your eyes anymore, and again, you're losing some of that muscle function. You usually become constipated, usually have abdominal pain, usually very dizzy. Again, there is a toxin that is now in your body. Usually within, if you still don't get treated whatsoever, it usually is deadly within about a week after eating something that was contaminated. So I always say, I'm like, don't eat a bulging can, because there was a reason why it's bulging. Something was growing in there. Another way that people pick up botulism, it's called infant botulism. And we can actually eat the Clostridium botulinum bacteria, and our immune system can recognize the bacteria and it can get rid of the bacteria because we have strong enough immune systems. It's only when you ingest a lot of the toxin that our bodies suffer. But infants, they don't have a very strong immune system yet. So if they ingest even small amounts of the bacteria, their immune systems can't get rid of the bacteria, which means the bacteria will grow in them and it will produce toxins. And again, it becomes toxic and deadly. One of the top places that you see any warnings about it without actually saying why it's warning is in honey. If you ever look at your container of honey, because I encourage you to go home and do that now, on the back side of the honey, it will say, don't give to infants. And you're like, why? So honey in itself is antibacterial. Bacteria don't like to grow in honey. That's why if you ever get stranded out in the wild and you get a cut and you've got nothing, but you can find honey, put honey on it. Honey is antibiotics. It's awesome. However, because it's antibiotic, we don't do a lot of treatment for it because really nothing grows in honey except this guy. So Clostridium botulinum will grow in honey. And even if it has Clostridium botulinum in it, we can eat that bacteria and it will be just fine because it's not growing happily in honey. But infants, if they eat honey and it has Clostridium botulinum in it, their immune systems can't fight it off and it can be deadly. So that's why I'd always say is don't feed honey to infants on the back of the bottles. And that's because it could potentially have Clostridium botulinum in it, and their immune systems just can't handle the bacteria itself. A more rare is that this bacteria can get into wounds. It can be deadly. It will literally cause all the same symptoms as a foodborne botulinum. It usually just takes an extra day or two. So eventually that toxin is going to circulate through the body and will stop all your skeletal muscle contraction. But does anyone can? I was going to say, I'm like, oh, no one can. I just started. I can't say anything. I'm like, I never canned for the last few years. The interesting thing, the fun factoid, if in future you ever decide to can, Clostridium botulinum can survive boiling if it's in an acidic environment. So there are two ways to can. This is my fun random thing. There are two ways to can. You can boil can and you can pressure can. So water boils at 100 degrees Celsius And pressure canning usually gets it up to 120-ish degrees Celsius because it's under pressure. You can get a higher temperature. Pressure canning will always kill this bacteria. And so it is more advisable, but it's putsy. And kind of a pain because I've done it. And I'm like, so I always prefer boil canning. But boiling will not kill this bacteria. So one of the things that they do in like every recipe that you boil anything is you add lemon juice. That lemon juice will actually lower the pH enough that it's now an acidic environment, and now you'll have acid plus boiling. That will kill the botulinum. So if you ever wonder, like, why does it always ask me about lemon juice? That's why. Now, there are some foods that are naturally acidic. Most fruits are naturally acidic, and so they can always be boiled. But there are some foods, like if you ever can meat or can certain vegetables, they are not acidic, that you have to either pressure cook them or you have to add that lemon. Fun little side factoid on why we do things. Now, to diagnose botulinum, well, they look for all the signs and symptoms, and oh, did you eat something suspicious? Oh, you had someone's home canning and they didn't really know what they were doing. Treatment, because the toxin is deadlier than your immune system, is as fast as, targeting those toxins, they can give you antibodies against the bacteria. Those antibodies will target that toxin and neutralize it. And then they're going to give you some antibiotics to kill any bacteria that are still around. Best prevention, proper food canning, and don't give honey to infants. That's the top ways that people get it. Now, the Clostridium tetani. It's our last of the Clostridium, and it works also against the skeletal muscles, but in almost the exact opposite way of botulinum. So Clostridium tetani, which causes tetanus, it's not the bacteria that causes the issue, it's the toxin that causes the issue. It's a tetanus toxin. So in all of your skeletal muscles, they are antagonistic muscles. So if I flex my bicep, I'm not really flexing, it's sad. But if I flex my bicep, that means on the opposite side, my tricep is relaxing. And I'm like, because it's stretching as the other one is flexing. So as you're always telling one side of your muscle to contract, you're telling the other one to relax. Well, what happens is this bacteria prevents the signal to relax. So your skeletal muscles, once they contract, can no longer relax, and they will stay in a contracted state. So one of the very first muscles that is infected by this toxin are the muscles of the face. And your skeletal muscles will contract in your face, and they will stay contracted. And so you end up with what's known as lockjaw. The only reason your mouth opens is because you are relaxing the muscles in your face and allowing that jaw to lower. But if the muscles stay contracted, they will stay contracted shut. They will stay, your muscles will contract and stay in a contracted state, and they will spread throughout all of the muscles in your body. That literally your entire body will go rigid. It's what that little picture is, like that little poor little boy is like, all of the muscles are staying in a contracted state. The muscles can contract enough that it's been known to break bones in the body, that those muscles literally contract and snap the bones that are underneath them. Now, I don't even have how do you get it? How do you get tetanus? Everyone's like, rusty nails. They can cause it. This particular bacteria is naturally found in soil. So it has to get into an open wound. So if you're playing in the dirt and you now have an open wound and it gets into a deep enough wound, again, it's an anaerobic bacteria. It has to go into that anaerobic state. So if it gets into that wound, into an anaerobic state, that's when it grows, creates the toxin and spreads. Everyone's like, rusty nails, rusty anything. If something's rusty, it just means that it's been outside, it is exposed to the elements, and if something's in the soil and a wind comes along, and it will deposit on that whatever surface it is. So rusty itself does not mean that it carries the bacteria, but it does mean it's something that's dirty because it's exposed to the elements outside. And if you get a cut, you now have that open wound, especially if it's a deep wound, and can be anaerobic. But really, it's, if you're ever playing in a garden or digging in dirt or whatever, and if you've got a pretty deep wound, you got to make sure it's covered because it puts you at a really high risk of picking up the bacteria. So treatment, you're given antibodies, those immunoglobulins, against the toxin. You're also given drugs to try to kill any bacteria that are still present. And they can also then give you the vaccine and hope that your immune system tries to catch up with everything. However, I feel like I had that story. I'm like that. Yes, there was not a couple years ago that someone in La Crosse who was an Amish farmer ended up with this, and I think they had to put him in a medically induced coma. Our hospitals don't carry the antibodies, so they had to order them in. He survived, which surprised me, but he was in the hospital for, a couple months. But it's preventable. We have a tetanus vaccine to prevent it. It does usually require boosters of around 8 to 10 years, depending on how your immune system responds, how strong of immune response you have. So it's easily preventable. Now we're going to get to our last group of bacteria that don't make endospores. If I don't get to them all, I'll just tape the last little snippet. There are 4 different bacteria that are gram-positive, bacillus-shaped, and don't make endospores. The first one is Listeria. This one you may actually hear about because there always seems to be recalls on bacteria that have Listeria in it. And the main Listeria that causes human disease is Listeria monocytogenes. When we gram stain it, they're a coccobacillus shape. So they're not perfectly round. They're a little more, kind of smushy, and a little more ovalish shaped. How we pick it up? Eating contaminated food, drinking contaminated beverages, which is where we get all the recalls from. These bacteria, when they do get in the body, they are intracellular parasites. So they want to get inside of our cells. This is a picture of one of our cells that's living. It's getting inside of the actual, one of our own cells, and so it can even live inside of our phagocytes. Our cells that are meant to eat things, digest them, and get rid of the byproducts, it can get eaten, but then not digested. And so it will live inside of our own macrophages, which is great for it. I mean, what a wonderful hiding place is if you can live inside of our own cells. So it's good for it to start to reproduce. Its biggest virulence factor that allows it to cause disease is the fact that it is intracellular and it hides out inside of our own cells. It's really hard to get rid of that. Now, for people that have healthy immune systems, our immune system eventually can recognize that we have infected cells and get rid of it. However, this bacteria can cause a very deadly meningitis in fetuses and in immunocompromised. This is a bacteria that can cross the placental barrier. Very few bacteria do, but this is one of them. And so they're always worried with pregnant women of eating certain types of foods that are at higher contamination rates because it can cause a very deadly meningitis in a fetus. It's also, when there are recalls or outbreaks of Listeria, generally the only people you ever hear dying are usually elderly immunocompromised because it causes that deadly meningitis. Healthy, younger individuals, usually not deadly for. Now, one random factoid is because it is found in food and beverages. This particular bacteria could care less about temperature. It will actually grow in the refrigerator. It will even grow in the freezer. Normally when we put stuff in our refrigerator upstairs, I mean, it's not dead, but it just stays at the same level. It's not growing and multiplying in there. This bacteria could care less what temperature it is, and it will grow just fine in the refrigerator. And so just by refrigerating foods does not always prevent infections from this particular bacteria. When we diagnose it, it is a bacteria that does have a flagella, so we can actually look at it underneath the microscope and see it. It has a flagella just on one end, and it will literally look like it's spinning underneath the microscope. Treatment, we do have antibiotics that can treat it, so it's not untreatable. We just have to diagnose it. But the top foods that this bacteria likes to hang out in, and so these are the ones that you normally see recalls, it likes to hang out in sprouts, Brussels sprouts, soft cheeses. So usually a lot of it's like the white cheeses, the mozzarellas, the soft cheeses, the spreadable, the bries. Raw milk, I don't know why people even drink raw milk these days. Smoked seafood, lots of deli meats and hot dogs. So it's usually, this is one of the reasons that they do not recommend pregnant women to eat deli meats and hot dogs. That's also because of nitrate levels, but also because Listeria can survive even some of the salt and stuff they put in there. Our next bacteria, this might be our last one, and I'll tape the last couple slides, of bacteria that are gram-positive bacilli and they do not make endospores, are the mycoplasms. Now, these bacteria don't even have to live inside of the human. They can be found out in nature just fine. So they're the smallest free-living microbes that are out there. But what's unique is that their cell wall, they are gram-positive bacteria, but their cell wall is thin, almost to the point it's non-existent. And you're like, well, but a gram-positive bacteria has a thick cell wall. Yeah, when we gram stain, yeah, it should have a thick cell wall. These are unique bacteria, though. They are genetically gram-positive, but phenotypically they don't have that nice thick cell wall. And so these bacteria can actually change shape. So sometimes maybe they are a little perfectly round, but maybe they're a little more rod-like. Sometimes they're long and skinny or fat and short, whatever. They can change shape a little bit more. They're picky to eat and grow. And so if we try to grow these in a lab to help identify them, they usually have multiple growth factors that we have to make sure get into the correct media even to grow these guys. But they can grow and seem to cause the most infections on the mucous membranes of the respiratory tract and the urinary tract. And so these are bacteria that can cause a number of pneumonia and urinary tract infections. These are not the top causes of pneumonia and urinary tract infections, but they are a cause of pneumonia and urinary tract infections. It's pretty rare. I don't know the last time I've heard of someone getting a Mycoplasma infection, but they're out there. One of the most common mycoplasmas that causes disease that you may have actually heard of, especially if you had this as a kid or know anyone that has kids that has had this, is the Mycoplasma pneumoniae. Again, it's an unusual bacteria all in itself because it requires those different growth factors and it changes shape, that it causes an atypical pneumonia. So it's pneumonia that's not really like the other typical pneumonias. And it's atypical because there's no white blood cell response. I mean, you have an infection, you should have a white blood cell response. And it's a very mild pneumonia that it is then sometimes called, nicknamed, walking pneumonia. Meaning you can go about your day, no problems whatsoever, and you're like, I have pneumonia. It's walking pneumonia. It's because this particular bacteria is growing in your respiratory tract super slow, so you still can go about your day without causing any rapid infection. It's spread by nasal secretions, usually targets kids. And I'm like that, okay, kids are dirty. But it usually targets kids. They have walking pneumonia. They're just fine. They can easily diagnose it with an ELISA, and it is preventable. Good hygiene, because it's spread by nasal secretions. You know, washing your hands after you blow nose, things like that. Has anyone had walking pneumonia before, or known someone that's had it? Like, I've known several people that have had it or had kids that have had it. Again, it's like they're sick, but they're fine. They'll be just fine. It just, you know, usually hangs out for a while. I will tape the last couple slides. I wasn't sure if I was going to get through them or not, and I'll get those up this afternoon.